Good afternoon, everyone. Thanks for the nice introduction. Maybe in the beginning I point out that I'm an engineer and neuroscientist, and so I'm in the interface of these two fields, and still trying to also reach the astrocytes, the glial field. My background is mostly in studying excitability of cells and networks, both incorporating these neuronal and glial aspects, and synaptic transmission, plasticity, and such issues. But I'm not kind of expert or has any track record in cognition or brain disease, but in this talk I hope I'm able to give you like a little bit ideas starting from synapses up to cognition and brain disease, and particularly how astrocyte-neuron interactions might be involved in all these levels. So we have known about astrocyte-neuron interactions more than 100 years ago by studies by Ramon Cajal and also others. But for most of the time in 1900, like for the past 100 years, we haven't really had tools to address them in such a detail than the neuronal cells. There are many reasons for this, and we don't have time in this talk to go into details to them. But I just wanted to point out that already more than 100 years ago, Cajal was drawing neuroglial cells, these dark brown cells shown here that have relatively small soma, many thick processes, and then also many fine distal processes that surround pyramidal cells. In this case, this is, I think, hippocampal circuitry. And on pink color, you see a big astrocyte embracing a pyramidal neuron, and then an interesting case where twin astrocytes are forming a nest around a pyramidal cell. From these times, we've come a long way, and today we understand that human astrocytes are far more complex and larger than their rodent and primate counterparts. And this is mainly due to the fact that over the past 10, 15 years, we have developed new imaging techniques, and particularly new image analysis techniques, where we can kind of reconstruct the cells at this level of detail. So in this slide, you see a typical protoplasmic astrocyte that exists on, for example, cerebral cortex in gray matter. And here you see a human astrocyte, and you can compare to the size of a rodent astrocyte. On white matter, we typically have these fibrous astrocytes, and you can compare the sizes and differences between the rodent and human also in the fibrous astrocyte case. So we have started to understand that these cells are really branched. They have really many connections and huge connectivity, more connectivity actually than neurons. And as I said, this is due to the fact that now we can really do this segmentation and stacking and have imaging methods and analysis tools to see all these fine branches. So with all this branch structure of astrocytes shown green on this slide, we can conclude that astrocytes cover many vessels and neuronal dendrites. So the blood vessels are shown here on red color, and then the dendrites on gray color. This is an image courtesy from Corrado Calli, who used to work with Pierre Magistretti lab and is now has his own lab in University of Turin. From the work of Robich's lab in 2020, we have also learned that cortical astrocyte layering is distinct from neurons. So if you look at the developing system, the P14 animal, we can say that there are superficial, mid, and deep layers. So there are basically like 3 different types of astrocytes, and this is during development. When we approach the adulthood, we see that there are these 3 gray matter layers of astrocytes. They merge into 2 in adulthood, like in this P56 animal. in contrast, this white matter layer, layer 6 astrocytes, they are maintained into adulthood. And we don't fully understand what all this means, but clearly we understand that there are anatomical changes during the development and when you enter the adulthood. So, with all this morphological and anatomical knowledge that we have now, we can safely say that astrocytes are ideally positioned to control brain functions. And these aspects that I'm highlighting here with the gray color or black or gray color, like that astrocytes are involved in pre- and postnatal development, like initiating neurogenesis, structure the brain, and maturate the pruned synapses. So these aspects are well understood already for 10, 20, 30 years. Astrocytes are also very important, as probably many of you know, and you mostly think that that's their role, like energy metabolism, like supporting blood-brain barrier functions, or they control glucose levels and then provide nutrients to neurons. And most importantly, which is perhaps the most and best known role of astrocytes, is to provide this homeostasis and survival functions, like maintaining extracellular ionic balance and taking up neurotransmitters to make like new glutamate to the presynaptic terminal. So based on these functions, we have kind of, I think we have stayed on this level, and we haven't thought that this homeostasis, actually providing homeostasis, is really crucial for everything that happens in our brain. And I will have examples of those a bit later. We now also know that astrocytes are involved in almost all neurological and neuropsychiatric conditions like epilepsy, schizophrenia, Parkinson's disease, and dementias. What we have less evidence about is how astrocytes are involved in plasticity, learning, memory, and cognition, and particularly whether they are directly involved in synaptic transmission and synaptic plasticity. Something else than just maintaining the ionic balance and taking up neurotransmitters. So this is an interesting emerging field, and over the past 10 years there's been more and more evidence about it. And this is also where my recent emphasis and work has been together with Ausa Saderkene. So for this symposium, I collected 3 slides evidence about how astrocytes modulate the brain signals and brain activity. So here are 3 examples about how astrocytes modulate network activity, brain oscillation, state transitions, and sensory processing. And if you go to the internet, you will find more, but I have selected those that were kind of eye-opening to me. In one of these studies, Lee et al. Showed already 2014 that blocking astrocytic vesicular release in transgenic, awake-behaving animals reduced cortical gamma oscillations and impaired object recognition. But other forms of memory, like working memory and fear conditioning, they remained unchanged. Then Boskanger and Juste, in their very seminal work in 2016, showed that through regulation of extracellular glutamate, astrocytes can trigger a slow neuronal rhythm in the brain that is important, has been like previously shown to be important in sleep and memory formation. And then in their very fundamental work, lines from the group of Alfonso Aracu to 2020 showed that astrocytes really respond to sensory stimuli in a stimulus-dependent manner. So this sensory stimuli first increases neural network activity in the gamma rays, and then there is follow-up by astrocytes. So there is a delayed activation of astrocytes, which then dampens a steady-state gamma activity. And there is an indication like that there is constant interaction, and astrocytes are really crucial in almost all brain activity. Then if we look at examples about how astrocytes modulate cognition, behavior, and sleep, here are a couple of examples. 2017, it was shown that the deletion of this kind of one gene in astrocytes alters circadian locomotor behavior and cognition, and this is through kappa signaling. And 2020, there was new study that this kind of conditional deletion of astrocytic transporters again produced loss of prefrontal cortex dopamine homeostasis, first of all, and then defective synaptic transmission and plasticity, and further impaired working memory and behavioral flexibility. So prefrontal cortex is very important in kind of providing this behavioral flexibility. And also from the work of Halassa and Haydon, and they have also many papers after this 2009, but in their very first finding they show that astrocytes modulate accumulation of sleep pressure. There are maybe less evidence about how astrocytes modulate synaptic plasticity, learning and memory, but there are a few of them, and I took those that were one of the first ones. So again, from the Alfonso Araque group, Navarrete et al. Showed that astrocyte calcium signal is necessary for cholinergic synaptic plasticity. This is, I think, in hippocampus, if I remember correct. And then in the very fundamental work by Min and Nevian, they showed that astrocytes modulate long-term depression in developing mice. And of course, these studies, they don't tell us like how this happens, but from a lot of other publications, we can get a kind of idea that very complex signaling between astrocytes and neurons is happening. And they use this signaling to kind of reach both the neuronal synapses and then the vascular system. What is instead unknown is really that, like we know that there are these mechanisms and there are these activity profiles that somehow seem to be changing when astrocytes get activated or are functioning, but we don't know how they relate to each other. And this is where I see that we really need these multi-scale models in order to understand all this complexity. And here I should perhaps mention, for those of you who don't know, astrocytes are not capable of producing fast action potentials, so they don't have enough localization of fast sodium channels to produce action potentials, but instead they communicate with the calcium signals. And this is the messy part of this work. So from this first part, we can conclude that additional studies are required to better understand how astrocytes regulate brain activity and functions in awake animals. And here I should point out that most of our understanding about these mechanisms, these signaling mechanisms that contribute to calcium signal in astrocyte and then one way or another contribute to the brain activity. So those are obtained from in vitro cell cultures of freshly isolated astrocytes or from brain slices. And now we need to kind of relate that to in vivo work. So we have been interested in applying both bottom-up and top-down modeling methods to understand the role of astrocytes in brain activity and functions. And recently, we've been, as I said, very interested in looking how astrocyte-neuron interactions are involved in synaptic plasticity. And I will now next go through this study, and then we will look at, in a very brief way, a couple of other studies that we've been doing. So Min and Nevian show that astrocytes are crucial for long-term plasticity in developing layer 4, layer 2, 3 synapse. In their study, they stimulated in layer 4 and were recording from pyramidal cell from layer 2, 3, and then they were also imaging cortical astrocytes in this very same layer. And they could see that astrocytes really, like when they block astrocytes, they don't see anymore this decrease in the EPSP as is shown here. So this phenomena is completely blocked when astrocytes are taken out of this experiment. And this paper is very complex, but I tried to squeeze the main results in this slide and with these 3 items. So they concluded in their study that time-dependent LTD induction is dependent on endocannabinoid-dependent increase in astrocytic calcium signaling. And it's specific for this type. In another synapse, elsewhere in a cortex, it can be some other neuromodulator, but in this case it's endocannabinoid, so it's specific for this synapse. And then this TLTD also requires exocytosis of glutamate from astrocytes. And this is the controversial topic, like whether astrocytes release something similar way as neurons. We now know that neurons have this very fast synaptic release, but astrocytes is a bit different, but they seem to be releasing, so there is more and more evidence now for it. And as I said, blocking this astrocyte calcium signaling inhibited completely the TLTD in their study. They also followed using 2-photon time-lapse imaging calcium transients in single astrocyte processes, as you can see here. And what they did is that they followed individual astrocytes during the plasticity protocol and after. And they could see that some of these processes are indeed involved in this phenomena and others don't contribute, so they are doing something else. And they were able to show like that the calcium activity, so this raster plot indicates the calcium activity, not the spikes, but calcium activity in astrocytes. So those shown with green are really changed after the TLTD induction. So we took this data into our consideration and developed a hypothesis that astrocytes modulate long-term depression in developing somatosensory layer 4, layer 2, 3 synapse through complex calcium signaling mechanisms. So we have to see here that these studies, Min and Nevian, and then also later group from Paulsen, didn't really show like the underlying mechanisms, that there were a couple of hints there. But then we had to go to this huge literature regarding astrocytes in order to further understand what's going on in this synapse. So our synapse model ended up having 4 compartments. So first of all, we have this presynaptic layer 4 spiny stellate cell compartment, and this postsynaptic layer 2-3 pyramidal cell has 2 compartments, soma and dendrite, and then we have a fine astrocyte process as 1 compartment. And you see here that this is relatively detailed biophysical model, so more detailed than many of those models that we have. So it's a Hodgkin-Huxley-based models for both pre- and post-synaptic neuron, and then lots of mechanisms contributing to intracellular calcium is also modeled in this system. With this slide, I try to guide you through the main assumptions that we made to test our hypothesis. There are also some minor assumptions that are based on previous studies and published work, but these were really the main assumptions. The first assumption was that this postsynaptically released endocannabinoid 2-HG can activate astrocyte endocannabinoid receptors. So there is a link there, and this has been shown many, including Min and Nevian, in their study for this specific synapse. Then astrocytic calcium-dependent glutamate exocytosis has an effect on presynaptic glutamate release. So there is a link from astrocyte to release of glutamate in this synapse. And then the missing link that was very difficult to find, but there was some indication in the literature about it, is that how to link the glutamate exocytosis and the presynaptic glutamate release. And in this synapse, it seems that the message is delivered through calcineurin. And calcineurin is activated through the calcium influx in the NMDA receptor. So this is kind of the main assumptions that we based the model on, and then went on to test our hypothesis. The model is very complex, and many of you might ask, why do you need such a thing? Well, I guess later we will reduce this to have like less equations than 80 differential equations, but in order to understand what is really going on in this synapse, we had to do this. We have altogether 170 parameters, and either the equations or data for these parameters that we have in these equations. So it's taken from these 70 references that we went through. And it's I must say that it's the wonderful work of Tiina Manninen who is able to dig out these things. I say a few words about fitting the model parameters because it's always asked. I never tell about it. But I would like to point out that we don't use any computational optimization strategy, but we instead use the well-fine-tuned neuronal models and still kind of verify that we are on a current range, like considering the action potential waveforms, calcium signals and astrocyte calcium signals and so on. And then we integrated the model, and to our big surprise, with these validated model components, we only needed to fix a few parameters in the presynaptic terminal in order to be able to start working with the full model. And I guess this shows the value of previous work and this very detailed fitting based on data. So there are really good models already available that can be used in this kind of studies. So I just summarize what we got. We did many other simulations, but I think this is our main result, which shows that we were able to produce the experimental result, meaning shorter temporal difference between pre- and post-synaptic activity. And now we are talking about this x-axis, that we have like the different temporal difference between the post-synaptic and pre-synaptic stimulus. So it can be like 10 milliseconds, 50 milliseconds, or up to 200 milliseconds. So this shorter temporal difference between pre- and post-synaptic activity leads to stronger TLTD through the astrocyte-mediated cellular and subcellular mechanisms. So we were able to produce quite closely the experimental results that were shown mainly by Panerse and also in Min and Nevian. What can we do with such a model? So this model is wonderful in such a sense that we can predict the dynamics of molecular reactions. So the concentrations of all those variables and molecular elements that are responsible for this phenomena. And this cannot be done simultaneously in any of the experiments. So we can, for example, follow the endocannabinoid concentration, IP3 concentration in astrocytes, which is responsible for partly producing those calcium signals in astrocytes, then glutamate in different locations, like when it's synaptically released in the extrasynaptic locations, and then what astrocytes sees. And then all these currents and calcineurin concentrations and such things. So we can follow their dynamics with this model to further understand what is going on. I conclude the model that the fine sensitivity of TLTD to the temporal difference is achieved through complex molecular signaling. And our model now links this complex signaling at the pre- and post-synaptic and astrocytic sites to the electrophysiology and time window of TLTD induction. And the model predicts for the first time the dynamics of molecular mechanisms underlying TLTD in this specific layer 4, layer 2, 3 synapse. I've been looking at the other synapses, like in other cortical areas and hippocampus, and there is similar things going on, but they use a bit these different synapses use a bit different mechanisms and neuromodulators to do this, but similar things are also ongoing in other synapses. So what does all this mean? So to me it means that during this TLTD induction, when the synapse is trying to kind of unlearn, a single astrocyte acts as a delay factor for fast neuronal activity. And in a way we could consider that astrocytes help mediate the integration of fast neuronal sensory processing with slow non-neuronal processing to modulate synaptic properties. And I think this has been the misunderstanding of the scientific community, that if something is very slow, it doesn't contribute. Yes, it can contribute in a very smart way, like we have shown with this model. And we currently consider that this phenomena might be related to synapse pruning and sensory learning during development. But as I said, something like this is also going on in other synapses and in different developmental phases. We still don't know about what happens in the adult animal, but it's an indication that at least during development this is very important. And indeed, if you look at, for example, the work of Nicola Allen and Kakla Eroglu, so they have shown that the astrocytes have really a fundamental role in activity-dependent synapse pruning during development. And this might have some implications to neurodevelopmental disorders like schizophrenia or autism. And there are now many review articles about speculating about the abnormal synapse pruning and how it could lead to neurodevelopmental disorders and the role of astrocytes and also other glial cells in that. And I conclude that our work presents one of the first evidence how this kind of dynamically could happen through complex astrocyte-neuron interactions. So, in addition to this study on the role of astrocytes in synaptic plasticity, we've already been for the past 10 years starting more and more to look at more about single astrocytes, both in cell cultures, freshly isolated and slice astrocytes, and also astrocyte neuron networks, and also how to model these things. There is, currently it's probably more, but we looked at more than 100, actually 106 models. And with this study we concluded that if you compare to neuronal modeling, there exist many detailed models that are fitted against data for different types of neurons. But for astrocytes it seems that there are only two types of models. Those that are based on Höfer et al. Type of model, and then this a little bit simplistic Dian-Kaiser and Lien-Rinchel model. So basically all these models that you see in this slide, so they stem from two models. So one single color represents one type of model, and basically all the models is the same, parameter value is the same, and they try to explain with those models different aspects of brain activity and disease and so on. So we conclude that it's in a very beginning, this field, and it has not really developed. But still, I think these models are used to produce papers, but they are basically using exactly the same model, and it's not explained how they are changed to explain something completely different in the brain. I just briefly mentioned that we are modeling both cultured astrocytes and then in vivo astrocytes, and trying to understand, as I pointed out, that these associated calcium signals are very complex. So we try to understand through these studies that, first of all, how the complex morphology in vivo affects these astrocyte signals, and then also how the diverse mechanisms in astrocytes contribute to these signals seen in astrocytes. We are also doing modeling of astrocyte-neuron interactions in networks, mainly in this kind of system where we use cell cultures that have glia and astrocytes. We've developed a workflow how you do the measurement of network activity with MEA, recording techniques, and then pharmacology, data analysis, computational modeling. And currently we are also extending this work to kind of provide a module in NEST that you could also incorporate the glial signaling in simulation of large-scale networks. We also do mathematical model order reduction in order to kind of transfer all these astrocytic complexity to kind of like more large-scale simulations, but in this talk I don't have time to talk about it more. With Alain Destex and Federico Tessler, we have also looked at a little bit like how neuron-astrocyte interactions and brain signals, particularly fMRI. And here the idea is to simulate neuronal and glial functions and try to understand how they underlie both fMRI signal. So with this very brief tour to astrocytes and astrocyte-neuron interactions, I would like to conclude that recent studies on astrocyte and astrocyte-neuron interactions have gradually transformed the field of neuroscience, or are transforming the field of neuroscience. And these three items that I selected here are perhaps the most kind of eye-opening. So human protoplasmic astrocytes are morphologically very, very complex. I think they are more complex than any of the neurons. Well, I haven't compared the cerebellum Purkinje cell, but maybe they are more complex. A single astrocyte can signal from 270,000 up to 2 million synapses in humans. This is a huge number. You can just imagine what it means in terms of computation. And the responsiveness of astrocytes, like neurons, is strongly affected by sensory stimuli. And this is perhaps the biggest evidence that they are not only providing the homeostasis, but actively contributing all the time. Astrocytes, secondly, astrocytes contribute to the fast dynamics of neural circuits. And I think we have also shown this with our model. And dysfunction in astrocyte morphology or function can cause neurological and neuropsychiatric disorders because they are all the time involved. Then my third conclusion is that modeling the multiple spatial and temporal scales of astrocytes is in its infancy compared to modeling of neuronal counterparts. And then I would like to thank my group, particularly Tiina Manninen and also Ausa Saudarkiene for this second part and very nice collaboration. And maybe at the end I would also like to point out that throughout my career since 1990s, the discussions with these really prominent CLIA scientists, I wouldn't be here. So it's really important that the experimentalists and modelers discuss together and develop a joint language. And I wouldn't have done any of this work without views from these people, and they are not all. I just listed the most fundamental ones, and to kind of sort out the controversies in the field to understand how to do these things. So, thank you.